Welcome to Alzheimer Speaks Radio. I'm your host, Lori La Bey, and I'm so excited that you're joining us today. We are going to have a fascinating conversation as usual as we learn from people all around the world at all ages and stages of life. Stay tuned as we shift our dementia care from crisis to comfort.
Here we go. What you think about, you will bring about.
Well, hi, everyone, and welcome back to Alzheimer Speaks Radio. I'm your host, Lori La Bey, and I'm thrilled you are joining us today. We're going to be talking about some research when it comes to Alzheimer's and dementia, which is always exciting to learn about. Now, if you liked our opening music, it's called Clarion Call, and the artist is the Mark Arneson Band. So you can download that on any of your favorite music platforms. We are so appreciative of them letting us use their music. And for those of you that are new to Alzheimer's Speaks Radio, we're about sound information, not just sound bites. We like to have real conversations with real people so that we get a good feeling, big and small, what's going on all around the world. So if you're diagnosed or starting to feel you're having some symptoms, maybe you're a care partner, a business professional, an author, a music director, a movie maker, an advocate, a researcher, maybe you're a child making a difference. We like to hear from everybody. So please reach out to me and I would be glad to talk to you more about being a guest on the show. I always like to do a couple of shout outs. I'm really excited. We're going to do some movie screenings again of the film A Timeless Love. I'm going to be in Winona, Minnesota, April 7th. And April 8th, and you can register for the screening and talkback by calling 507-454-5212. That's 507-454-5212. And then I am still doing some support groups online. One is the Memory Cafe, Arthur's Memory Cafe. We meet the second and fourth Wednesday of each month at 1 o'clock Central Time. Anybody in the world is welcome to join us on that. And then also typically in person, we meet the fourth Wednesday of each month for Brookdale North Oaks Caregiving Program. And hopefully this next month coming up in March, we will be able to meet in person. Otherwise, we will convert to virtual. Also, I want to mention the Dementia Map. If you haven't checked that out, it's a global resource directory that is building out more and more every day. There are 150 resources you can check out on the resource directory itself, but there's also a glossary of terms, there's a blog, and there's an events calendar. Now, we are going to hear from the Adaptive Equipment and Caregiving Corner, who has done a wonderful summary of the footbar walker. And I think you're going to want to hear this, and we will be back with our guests.
I love the footbar walker, and let me tell you why. It is the option for my toolbox that I've been waiting for. Let's be honest, there are some clients who, despite our best rehab efforts, just aren't able to return to performing a sit-to-stand transfer on their own. Now I can offer my caregivers an easier, safer option that doesn't involve hoisting their loved one up from a sitting position. I don't recommend this walker for all of my clients, but I do recommend this walker for those caregivers looking for an easier, safer option with transfers. I would also encourage other therapists to add this walker to their toolbox. It's kind of like having my own mobile parallel bars for the client to pull up on. Whether it's a family caregiver at home helping a loved one with Parkinson's or dementia, CNAs in a long-term care facility assisting their patients, or therapists adapting to client and caregiver-specific needs, we now have a very safe and effective option to offer in the footbar walker. Check this product out at thefootbarwalker.com. That's it for today from Adaptive Equipment and Caregiving Corner. Have a great day, and don't forget, if you can't do it, adapt it.
So I am really excited to introduce our guest today. We have Maria Maccecchini with us, and she is the founder and president and CEO and executive board member of Annovis Bio. She has devoted her life and 30 years of her career to studying neurodegenerative diseases and their possible treatments. In 2008, she founded Annovis, and this is her second biotech company. In addition, we're going to be talking with Cheng Fang, and she is the Senior Vice President of Research and Development at Annovis Bio, and she is an accomplished neuroscientist with more than a decade of experience in neurodegenerative diseases with a broad scientific knowledge and hands-on experience. Well, ladies, I am thrilled to have you join us, and I can't wait to learn more about your work. So thank you so much for taking time out of your day. To start, I always like to ask every guest if they have been personally touched by dementia in their own family or circle of friends. And Maria, I'm going to throw that to you first, if you don't mind.
Sure. Thank you for having us. Yes, in fact, my mom died of Alzheimer's a few years ago. Her sister died of Alzheimer's a few years earlier. And while that seems like I should be scared, I'm not. They were both way in their 90s when they came down with the disease. And realistically, unfortunately, by the time we are 90, all of us, we have a 50% chance of getting Alzheimer's disease. So the fact that they got it this late, I almost have to be thankful for it.
Yeah, I would tend to agree. My mom had symptoms starting in her 50s and lived for 30 years with the disease. So yeah, later I think is better, especially if it runs in the family, you know. And like you said, with aging, most of us are gonna be dealing with something like that. How about you, Cheng?
Yeah, my grandfather from my mom's side got Alzheimer's disease in his late 80s and died of it about a decade ago. So
Okay.
prevalence these days, it's almost hard to find someone whose family, either direct family or indirect family, not touched by those diseases.
Very true. Very true. I've spoken to groups of 1,000 people, and I'll ask like 6 questions. I'll have everybody stand up, and then I'll say, you know, has your grandma, your grandpa, and they sit down one by one. And there might be a handful of people out of 1,000 people that haven't been touched, and people look around and they're just shocked because we still don't talk about it for the prevalence that it exists. And so, you know, we need to get that door open to more comfortable conversations. Maria, I want to have you talk a little bit about how did you get into neurology, you know, and what was so enticing about that career choice for you?
I think I got interested in the brain way before I knew that it was called neuro something. So I was always fascinated by how things happen. Very early on, I became interested in the development of termites and of cockroaches, and when their brain decides that they become adults and can make babies, and when their brain decides that they cannot do that and have to remain infertile forever. So I thought that was fascinating. And in fact, I would open brains and look at these different brain pieces. And if you hit one, you make their development go in one direction. And if you kill the other piece, you make the development go in the other direction. I thought that was fascinating.
That is fascinating. That's very fascinating. How about you, Cheng? Was there something that pulled you into this realm as well, or?
Yeah, I actually came to the US for grad school for oncology. So in the grad school, they usually have you rotate 3 different laboratories so you can choose which one you're most interested in. So the first one I went into is actually using Drosophila for a neuroblastoma model. So I got very interested in neuroscience. I actually took more courses in neuroscience and joined a neuroscience laboratory for my PhD work on anxiety and depression. So I never looked back.
Okay, well, I'm glad both of you haven't, because you're doing some neat things. Cheng, there's just seems to be this huge failure rate out there in terms of development for Alzheimer's therapies. And I'm wondering if you can tell us why you think that is, and what are we doing wrong out there?
It's just so hard. So usually if you compare it with oncology, oncology usually is very gene oriented. So you have a mutated gene, so you know where your target is. But for neuroscience, especially if you look at Alzheimer's, Parkinson's, ARS, we don't really know what are the genes linked with it. And we don't really know exact the cause of it, except for age is the most biggest correlation with any of those neurodegenerative diseases. So second, if you ask the scientist, then yeah, they will have to start with animals, right? You can't just start with humans. Not having a direct good animal model also contribute to that, right? Because we mimic what happens in humans first in animals. And then if it's efficacious in those animal models, we have some indication we can test it in humans. So all of these, because of lack of understanding, we don't really have good animal models. And third is in the brain. So it's really hard to penetrate the blood-brain barrier. So that itself excluded lots of small molecules. And then the last but not the least is you are treating people who, even though diseased, but can still, live for decades, right? So you cannot be like oncology, put people on radiotherapy, chemotherapy. Both are extremely toxic. But when you look at life or death situations, you can let go a little bit of toxicity. But for neuroscience, we have to be extremely cautious. We don't want to do any harm to these people beyond what they're already suffering.
Yeah. Well, I have to say it's refreshing. You are one of the few researchers out there that I've had on air that says we really don't know. And that's the belief of the general public is we really don't know, but we seem to kind of keep tunneling down one path over and over and over, even though there's been some evidence that, hey, this might not be accurate. And I know personally for myself, I just think it's such a baby disease that we know so little about it. And, new phraseology and new terms are popping up all the time in terms of the different types of dementia that are out there. And yet everyone still, for the most part, still refers to it as Alzheimer's disease. And yet there's so many differences out there. So, Maria, I'd like to ask you, you've been 30 years in this career. And, what have you seen in terms of major changes in research? Has there been any?
Well, I think the major changes have been in cancer. When I was a little kid, there was nothing, and then there was chemo, and then there was radiation. And while they work, they're pretty toxic, as Cheng just said. And now we have immune oncology, which is very specific for one cancer drug. And that is really where we need to go with Alzheimer's disease, not necessarily with a neuro approach in terms of immunology approach or antibodies. It could be a small molecule. And I feel that the next 10 years are really going to be the 10 years where we open up a lot of different research avenues and development avenues and come up with a lot of new drugs. In fact, I should say we have been coming up with a lot of new approaches. If you look at the small companies that today all work in neurodegeneration, every single one of us has a different approach. Now, we don't know which one works, but there is a good chance that a number of them will work. And if we have a few drugs that finally work, then we can start doing combinations, we can start looking at effects, we can start subdividing the disease and figuring out, as you just said, different dementias may need different treatments. So I think the next 10 years will be very exciting for the area, whereas for the last 20 years, we have been pretty stuck.
I've had a lot of people comment, with this latest treatment that was out, and then it was kind of, said, nope, we're not going to approve it. And then it came down half in price. And people are saying the whole process that happened there, they feel is going to harm future therapies. So I would love to know what both of your thoughts are on that. I'm going to go to Maria first.
it's hard to decide what's going to happen. You can have both versions. You can say it's a small step. The drug works a tiny, tiny little bit, but it might help a tiny, tiny little bit. The drug has a lot of side effects. Is the efficacy worse the side effects? And you can decide yes, you can decide no. You can say it's the first step in a long trip that we have to go. So exactly how that's going to impact us, I cannot know longer term. Short term, it has really dropped the stock, because if a large company can't figure it out, why can the little companies figure it out?
Mm-hmm. Yep. Cheng, anything that you want to add on that?
Yeah, I actually think, tau hypothesis, A beta hypothesis has been two prominent hypotheses in Alzheimer's field for decades, right? At least the past 20 years. So they have their reasons why they are very appealing hypotheses. I think for the antibody drug, for what's worth, it actually works beautifully in cleaning plaques in human brains. So I think, but then it has very little, if any, clinical efficacies in patients' outcome. I think it tells us very clearly, this hypothesis may not work or may not represent the full picture. So I hope the field would learn and actually pay more attention to other alternative mechanism of actions, which, like Maria said, actually, if you look at the little companies, we're all working on different mechanism of actions. So I think that would be the trend for the next 10 years.
And I have to say, I talk with people all over the world at all different levels, and the excitement for the smaller companies is pretty high, because they feel like there isn't one track that's going to get to the bottom of this, and they want to see a wide variety of therapies being pursued. And they see so much overlap between their disease process too, depending on which one they have, or some have multiple, combinations of dementia and stuff too. So Maria, can we talk a little bit about your company looking at how do you reverse some of the neurodegenerative targets, with these neurotoxic proteins that are in our brain, and give us a little bit of background with that?
Yes. So as Cheng just said, and most people know, the main targets is A beta and tau, plaques and tangles. If you look at the brain of an Alzheimer patient, there are also Lewy bodies that are actually usually put into Parkinson's. And there are TDP aggregates that right now people thought were only in ALS. So as you keep looking, you see more toxic proteins. So if you just take away one, that's really not good enough. You may get a small effect, but you still have the other toxic proteins. So how does our drug remove more than one? And it just so happens that nature designed these proteins to be regulated the same way. So their translation, their manufacture is done the same way. And because their translation is the same, we can inhibit the translation of all of them. If it wasn't the same, we would have to inhibit each one independently. But for reasons that nature decided, and there are some hypotheses why that is so, they all are regulated the same way. And so by interfering with that regulation, we lower all levels.
Well, that's really interesting, and that's really exciting too, when you've got those multiple variants, because we hear, how expensive this whole process is. And, if you can get everybody on the same train track with one thing, that's pretty exciting. Cheng, what about you? What do you think the mode of action needs to be in terms of pursuing these proteins?
I think, like Maria said, our drug has a very unique mechanism of actions, right? So this, it took us a while to figure this out, and it works beautifully. It actually doesn't work in a normal cell situation. It's only when your cells are challenged, injured. So it would produce a lot of ion inside. So that would activate the overexpression of those neurotoxic proteins. So then our drug would come in and lower the levels of all those neurotoxic proteins from APP, which is a precursor for A beta oligomers, and tau, and TDP-43, and prion, all these aggregating neurotoxic proteins. So in that way, we don't just remove one, so then the other neurotoxic proteins still are playing, activated the bad molecular pathways and injuring your neuronal functions. We now remove all the bad guys. So in this way, it can have much pronounced protective effects.
Okay. And what's the name to this, transitional inhibitor?
Oh, we just got our generic name. It's buntanetap.
Okay.
My first choice, but no choice.
Okay. Well, that's good. I thought I would probably crucify pronouncing that one. So I thought I'm just going to ask you to do it on that one. Maria, how is this drug able to really inhibit more than one? I mean, is it, I still don't understand all the makings of how it targets them. Is there a way to dumb it down for me?
Yes. I'm not sure I can dumb it down, but I can try to explain it differently. So, as you know, DNA makes mRNA. And in the meantime, we all know mRNA makes protein because we have it in the COVID vaccine. So mRNA makes APP, makes tau, makes alpha-synuclein, makes all the things that are toxic. Well, mRNA needs to go to a ribosome to become a protein. I mean, not to become a protein, to be translated into a protein. So the mRNA has the coding to make a protein, and there is this big protein called the ribosome that when the mRNA comes through, reads the mRNA and makes a protein. It's beautiful. I don't think we could have invented it. It just is really beautiful the way nature works. So, okay, so you have the mRNA and it needs to go to the ribosome, and the ribosome makes the protein. What our drug does, it keeps it bound to something else. So it doesn't go to the mRNA. There is another protein out there, and our drug binds it to that protein, so the mRNA cannot escape and cannot go to the ribosome. And therefore, the toxic protein is not made.
Okay, because I was wondering, does the body flush that through then? If it doesn't, if it's not able to become toxic, does it flush it out then?
Well, that's an excellent point. See, we talk about toxic, but the truth is A beta, tau, and alpha-synuclein have a function. At normal levels, they're normal proteins. They do their job.
Okay.
They do their job in terms of signal transmission. They do their job in terms of having microtubules. They do something. But the minute you have too many of them, they get toxic.
Mm-hmm.
Because they become clumps. You have heard that some of the antibodies that work against oligomers. Those are small clumps of these proteins. And then they make big clumps of these proteins, and then they're called plaques or tangles.
Okay. Cheng, anything that you want to add to that?
No, I think Maria did a pretty good job in explaining this. Yeah, it's pretty complicated a mechanism, but just in short is our drug by preventing those neurotoxic proteins, MRAs, being translated, so we can regulate them at a normal level, right, to maintain their normal functions. In that way, we can preventing them being overexpressed and form those clumps, and therefore we can preserve the normal cellular environment and preserve the neuronal functions.
Okay. I guess when I hear the term toxin, I always think, well, get it out of me then. Just flush it on through, and my mind doesn't think of it's just in overdrive. It's too much. It's,
But Lori, you're 100% correct. If we made these toxic proteins in the tongue, for instance, we could just kick it out. Mm-hmm. Tongue regenerates, saliva takes them away, it's the end of it. If we make them in the brain, where do you put them? We have a skull. We have a blood-brain barrier. The brain is totally enclosed. They can't go anywhere. And in fact, some of the approaches help them come out, including sleep. People have found that if you sleep well, you clean up the brain better. So you were correct in saying, let make them go out. Problem is the brain can't do that very well, other than if you stay in bed all the time.
Well, and we have been hearing more about getting enough sleep and what that does to regenerate and cleanse the brain as much as just stabilizing us and giving us energy as well on that. Let's talk about, are you doing any clinical trials at this point, at your company?
Well, we just finished 2 phase 2s, one in Alzheimer's, one in Parkinson's. We did summarize our Parkinson's data and send it to the FDA and asked if we could go into phase 3. And they cleared us for 2 phase 3 studies, one in early and one in advanced Parkinson's. We plan to start the first one of those 2 studies as second quarter in May. Okay. With Alzheimer's, we haven't quite gotten there yet. We are still putting all the data together so then we can go to the FDA. And we expect to also do some additional studies in Alzheimer's disease. But for Parkinson's, we're pretty set. Phase 3 starting in May for the first study, then sometime later in the year for the second study. And somewhere in between, then we're going to start the Alzheimer's study.
So do you know yet in terms of the Parkinson's study, how that'll come about? Is it in certain locations within the globe, or, how does that work?
Yes, we are in Europe and the United States, and it's 100 sites in Europe and in the United States. It's 450 patients, 3 times 150, and it's 6 months. Okay. And can you tell us, in terms of the study, what someone would expect if they participated in it? Cheng is my Parkinson's expert.
Okay.
Yeah, it's pretty simple. We try to make it as low patient burden as we possibly can. So basically, it just involves patients coming first time for screening, right? Our criteria are pretty straightforward. If you are early Parkinson's patients, which is defined by a whole and yarn score that your neurologist would know, and then it's criteria 1, 2, 3, and then I think that's pretty much about it. We would attest to your cognition in the normal range. That means you don't really need a caregiver to come all the time with you and take care of you at home. So that's our very simplified inclusion criteria. And then you come in once a month for 3 months, and then have a phone call in between. And then the 6th month, you come into the clinic for the end of trial. That's about it. We do not collect any of the spinal fluid. We do not do any of the expensive imaging. So we just take blood samples for both biomarker and also for our pharmacokinetics requirement.
Okay. And so then would they be taking a medication on a daily basis then, or?
Yeah, yes, absolutely. Once a day, easy.
Okay. And is it a double blind so people don't know if they're part of the study or not? And
in the meantime, they call them quadruple blind.
Okay.
It's still a double blind, but they make sure it just specifies that the caretaker, the family, the nurse, nobody knows.
Mm-hmm. Okay.
It's totally blinded.
Well, interesting. And so with the Alzheimer's trial, the question that I have, and I think a lot of people do too, is with these studies, how it's defined, because so many people say, well, they said I had Alzheimer's, but now I have this, now I have this, or, and so they don't feel, a lot of people don't feel confident in that diagnosis that it's really, truly accurate. So how do you handle that? I mean, I suppose you have to go by the definition that is currently out there and what people have been diagnosed at, but it just seems like there's a lot a lot of people, and again, I talk with people all over the world, this seems to be pretty consistent where their diagnosis changes.
I feel a lot of it is psychological. Sorry if I'm a little nasty.
Mm-hmm.
But my mom had Alzheimer's. I called it Alzheimer's. Her friend, who was 2 months older, her daughter refused to call it Alzheimer's. They looked the same. They behaved the same. Her daughter insisted it was vascular dementia. Now, honestly, does it make any difference?
Mm-hmm. Okay.
So, but the word Alzheimer's didn't want to go through her mouth.
Mm-hmm.
So she insisted. I said, did she have a stroke? No. Well, we don't know. Yes, but does it make any difference? So on the other hand, if we had a drug that was specific for vascular dementia, then yes, then it's important that you make that distinction. But at this point, we don't have it.
Yep.
Also, what our drug shows, that's why I want to develop it in Alzheimer's and in Parkinson's disease. It doesn't care too much how you got to having diseased nerve cells. If you do have diseased nerve cells, it's going to fix them. So whether you have vascular dementia or whether you have Alzheimer's, our drug will probably help.
Okay. And, from what I was saying doctors were changing the terms, not so much, but I have seen families that have refused to use certain terms as well. But, I've seen so many people who were living with Alzheimer's for many years, and then all of a sudden, no, now it's cognitive, mild cognitive impairment. And they're saying is.
But Lori, mild cognitive impairment is very early Alzheimer's.
Mm-hmm.
You see the difference?
Yep.
I found a new word to make people feel less bad.
Mm-hmm. Yeah, well, they got more upset over it because they said there's nothing mild about this.
Okay, I got it.
There's but it, or all of a sudden it's Lewy body or it's frontal temporal or, whatever. And again, I think we just need a lot more education out there too with some of our doctors. Not everybody is trained in this. Plus, I know it's fluid. It's changing all the time. When my mom had her autopsy, we were told, well, the first 10 years she was told it was just hormones, and she knew it wasn't just her hormones because it was very different from her girlfriend's symptoms. And then they finally got the Alzheimer's diagnosis. And then upon death, we did the autopsy, and it showed that she had Parkinson's and a little bit of Lewy body. And, the neurologist asked, did you see anything? And I said, well, from the Lewy body, we didn't really see any hallucinations or delusions. And I said, well, I suppose maybe, she'd be sitting there sometimes talking to people, but it was almost to me, and some people won't agree with this because they'll see it as a spiritual thing, where she had kind of crossed the veil and she was talking to people that were past, but she wasn't upset. She was comfortable in those conversations where I hear so many people are frightened by their dreams and those types of things. And then with the Parkinson's, it was really hard because she had a little bit of a tremor, but then she ended up being wheelchair bound. And then you don't see as much, of some of the symptoms and things there. So it is kind of a crapshoot with this, but it's nice to hear that you feel, your trial, can deal with all of those things, because sometimes people, I know when they're signing up for a trial, they're just not sure, do I have to have that diagnosis of Alzheimer's to be part of the trial, with that? So that would be a question I'd ask. If somebody had Lewy body or frontal temporal or vascular, would they still be able to be part of the study? And I know you're just submitting it at this point. And
that's an excellent question, because let me go just a tiny little back. You said your mom had Lewy bodies, and she probably had dementia of Lewy bodies.
Mm-hmm.
Kind of looks like Alzheimer's, but it's caused differently. But do you remember in the beginning, Cheng and I said that in the Alzheimer brain, there is a beta tau alpha synuclein and TDP43? Turn it around. In the Parkinson's brain, there is alpha synuclein, tau A beta, TDP43. So it's not a clean disease. That's why we have such a hard time getting drugs to work by working on one neurotoxic protein only. It's not a clean disease. And different manifestations may have very high TDP-43, very low A beta. Doesn't mean they don't have it. It just means reversed. So that, and the reason doctors change is that there is a new imaging mechanism. So they use the new machine and it tells them this is what's there. But the truth is there are probably 4 things there.
Yep. Cheng, I wanted to ask you about, past trials in terms of what you've observed too, in terms of biomarkers and outcomes and also patient reaction to being part of the trial.
Yeah, that's a great question. So let me first answer patient reactions. It's quite interesting. So Parkinson patients, even though it's a quadruple blind study, they usually would feel the difference. So even in the trial, so they feel they're better. And some of them would write to us, write to Maria, and saying that, hey, your drug works. And then she would be very confused because she didn't even know what the patient was taking, placebo or our drug. So it's only after, we close the blind, which means all the data are sealed and no one can change anything. Then we take a look at these patients. Actually, indeed, all the patients were told her that, hey, I feel better, are taking our medication. And some patients were complaining, your drug don't work, we're on placebo. So that's very exciting. I think it's because the movement changes are easier to spot and easier to feel an improvement rather than a slight improvement of your cognition in Alzheimer's patients.
Mm-hmm.
So we're quite encouraged by that. Actually, we recently went to a boxing gym just for Parkinson patients. So some of their patients went on our previous trials and were told other patients about it. So they invited us to give them a talk about our upcoming trials. So for biomarkers, it is, we did both CSF biomarker and the plasma biomarker. And we did a lot of the biomarkers in this 2 phase 2 studies. So the reasons are twofold. First is the biomarker usually are used to validate your mechanism of action, right? Because, for patients, especially in the brain, we can't really open your brain to see what's going on. We can use the little protein levels in your blood, in your spinal fluid to try to figure out, what is going on, whether it's targeting the proteins that we wanted to target, whether it's targeting the pathways we wanted to target. Also, we're doing both is because, for doctors, if you say, hey, you need to do spinal fluid, they're like, yeah, sure, the patients are used to it. But still, when you think about it, it's a lot of a burden to the patients' families. So we wanted to remove that. So in this trial, we kind of to validate the biomarkers we wanted to see, comparing it from plasma to CSF to make sure that next time we don't have to do the CSF.
Okay, great, thank you. Maria, is there anything that you wanted to add to that? And then I've got another question for you.
No, I think Cheng said it very well. My biggest concern with patients is they're suffering. You really don't need to stick them in a study where you stick them all the time, where you stick them into machines, where, you do the best you can to keep it to a minimum and to make them comfortable.
Yeah, I agree. There's so many studies that people struggle with, and sometimes it's just even in the process of getting into a study, jumping through all these hoops and then getting kicked out at the last minute that really frustrate people. I found it interesting when you were talking, Cheng, about you got invited to the boxing club because people were there, and I've just heard so many people with Parkinson's talking about the boxing and stuff. So it's nice that the word is spreading on the ground in terms of the studies in and of itself. Have you guys had problems getting people into your studies? Because I know that is something I hear from a lot of different companies.
Not for Parkinson's. Parkinson's patients are younger, about 10 years younger than Alzheimer patients on average, and they're still fighters. Their brain works usually quite well.
Mhm.
So because, we really don't do that much. Taking a pill a day is not a major issue. You take probably 150 pills anyway, so one
Mhm.
more is going to make any difference. So no, Parkinson's, we were very lucky. I foresee that in Parkinson's, we're going to be done very fast. Alzheimer's a little harder, mostly because a lot of the people with Alzheimer's stopped driving. So
Mhm.
they do need a caretaker. So it's more complicated. Then they're older. Like in phase 2, we took spinal fluid. As Cheng said, we don't want to do it in phase 3, but we did in phase 2. So if your mother is elderly, has some comorbidities and has Alzheimer's, would you tell her to take 6 hours of spinal fluid? And it was mostly the daughters who said no. So
Mhm.
it was a little harder to recruit Alzheimer patients.
Yep. Yeah, it definitely is. Well, Maria, what's the outlook for the company? Where do you go from here? I know you've got this trial for Parkinson's that's approved, and you're getting that up and running, and you're working on the Alzheimer's trial. What else is down the road for you?
Well, I don't think that more than four clinical studies in phase 3 are down the road for me. I think I really want to prove, this has been kind of my life, that my drug protects nerve cells from dying, okay?
Mhm.
And I think it does, okay? All the data I've seen to date says yes. So now we're going to show it in phase 3. You can't do more than phase 3. If it works in phase 3, we're done.
Well, that's pretty exciting. So for the trial itself, you were talking, I think, was it in May that you
Yeah.
were going to start? And then you've got 6 months out. And then how long does it take you to gather and then push everything back and go, approve
It's
us?
not 6 months. It's 6
Uh,
months for 450 patients.
okay.
So you don't recruit them all at day 0.
Okay. Makes
Recruit
sense.
them over time. So it's going to be 1.5 to 2 years.
Okay. Okay. And then to like analyze all the information and then kind of
But
package that.
that's a few months. I think that the study really, in the beginning, I believe we're going to be fast because,
Mhm.
with these gyms, if we spread the word, they're eager to participate. We show them the data. We have excellent phase 2 data. So, while the study is blind, they're not blinded to the fact that this drug worked in a different study. Okay. So I don't see any problem in the beginning. But then the sites, they run out of patients. It's going to get slower as time goes on. Yeah, 1.5 years and then another few months.
If COVID kicks back into gear, with another variant, which I know there's a couple hanging out there, do you see that as slowing the process down?
Not really. I mean, we did CSF in COVID, and in the beginning, it really slowed us down by about 4 or 5 months. And then slowly, you pick up a lot of safety procedures. It worked pretty well. We had one COVID case in 100 patients.
Okay.
That had nothing to do with us. He got it from his grandson. But the fact is, he did get COVID. So, no, I don't see a problem, again, for Parkinson's. They'll be fine. I may see a problem for Alzheimer's because, again, the people are weaker, they're older.
Mm-hmm. Okay. Well, and people are getting used to, the rules and the processes, and they're tired of being cooped up. So they might be more likely to step out, where in the beginning, it was just scary, so scary for everybody. And yet we all thought, well, this will be over in 2 to 3 months. And boy, we were wrong on that one.
3 years, yes.
Well, good. Now, is there anything that we missed any questions I should have asked you that I didn't, Cheng?
No, I just want to add on to what Maria just said. We actually also learned from the previous trials. Since our trial design is really simple, so if really comes down to that, another round of lockdown, it's very easy for us to do what we need to do at patient's house. The simple tests that need to be done can be done through Zoom And the blood sample, we can send nurses to come to your house to collect because it doesn't need any expensive instruments. So I think we're well prepared and be ready for the next trial.
Okay, great. Great. Now, if people want to get more information, they can go to your website, which is annovisbio.com. That's ANNOVIS and then bio.com. And if anybody out there has any media inquiries, you can go to Russo Partners, LLC, and you can ask for Erica, ERICA dot, and then last name is FIORINI at Russo Partners LLC. So that's RUSSO Partners LLC.com. I really thank you both, ladies, for your time today. It was really interesting learning of what you're doing and exciting. It was uplifting when we hear so much kind of doom and gloom about, therapies that are out there. It's nice to hear that, something's brewing that looks pretty dang good out there. So thank you so much for your time.
Well, thank you. Yeah, thank you for having us.
Hi, everyone. This is Meredith from the Senior Fitness with Meredith podcast, where I discuss all things for seniors, from fitness, your health and wellness journeys, how to be all over strong and beyond. I also have my mini podcast called Motivation with Meredith. It's a great, quick motivational pick-me-up for your days. Join me, listen now, search for Senior Fitness with Meredith on your favorite podcast platform.